and Brian have all collected samples from compost and soils, and they're going to do a deep dive and search for some of the organisms that they can see in the soil. And they're going to explain a lot about what they're seeing, give you a bit of context, get you a little bit of intro about whether how you can, identify this yourself in the soil. And yeah, let's just kick this off. Who would like to start?
We didn't meet about it. Brian, do you want to start with the boring stuff and build up? Because I added water to my organism and it seems to be just
Sure. Yeah, I'll start actually. I'm going to start with my soil from my fruit trees. They're actually doing fantastic, but lo and behold, there's a root feeding nematode. So if you guys want to see a root feeding nematode, we'll take a look at that first. All right, let me go ahead and share out here. All right, let me know when you guys can see.
Coming. There you are.
All right, let me put
Nice.
that
Yeah.
down. Okay. Can you guys see it all right?
Yeah.
Actually, yeah, let's see if I could go. There was a way to go full screen with this. Well, let me zoom in. Let me just do that.
Is it like view? Do you go like view full screen in tube view?
Yeah, I think that might be in there. View full screen. Thank you, Raleigh.
You got it.
There we go. Can you guys that
Nice.
look better?
Yeah. And
Better.
this is live, everybody. You're seeing a
All right,
live
so
sample.
context here. So how do we know that this is a root feeding nematode? And I want somebody in chat to tell us what makes this a root feeding nematode. I know there's some people out there that know it.
I, I
Casey's trying to raise his hand. Calm down, Casey.
it's me.
Did anybody get it right?
Yeah, mouth
Oh,
parts,
oh,
spear in the
oh
knob,
yeah, we got some good answers. We got some good answer. We got
mouth.
Astrid
Yeah,
says knobs, Isabella says the mouth, Gordon says looks like a spear, and Dan says spear too. So we got
right
some
there.
smart cookies here.
Yeah, and so let me just give you a little context. You don't always have to freak out every time you see a roofing and nematode. So this is coming from an Asian pear tree that I have in my front yard, and I tell you what, this pear tree is doing fantastic. Super vigorous growth, great fruit production this year. The leaves are looking fantastic. Really, no pest or disease kind of indications that are showing on this plant. And lo and behold, what do I find when I'm looking through the soil? I find a root feeding nematode. So they're in our soils for sure. Does it concern me? If I had no context, I might look at this and go, "Ooh, I might be a little concerned." But knowing where this came from, then I'm knowing that this is not actual, a problem for me in this case. If I was seeing lots of root feeding nematodes, then yeah, I'd say, hey, maybe I have an underlying problem that I need to deal with. But in reality, it's just kind of cool to look at, and I'm not too horribly concerned about this.
Hey,
All
Brian,
right.
can we talk about
Yes,
that a little bit more? Because it came up in Compost Academy this week. Megan had a really good question. I'm
sure.
fairly certain Megan's not here right now because of her awesome job traveling and looking at soil biology all over the US. But
Sure.
she was talking about how she had a client recently that she was assessing the soil and
Yeah.
she saw that the soil itself looked pretty good. It had nice structure. They didn't go down farther than, say, 6 or 8 inches. The client does till, I think, annually, once a year. And and the client's really happy with their crop. When she looks at it under a microscope, she's saying, I'm not seeing the entire soil food web in place. How is it that you could have soil that's not compacted, that's growing food that the farmer's happy with, and not see all the players in place? And I wrote my answer online, but I'm curious. I wanted to hear what you had to say
Yeah.
about
Sure. I read your response, and I'm in that same boat, which is, yeah, even though the soil may not be wonderful, like when you see a really good soil, you see it really well aggregated, the huge component of the organisms to make up the soil food web, and you know things are functioning correctly. But that doesn't mean that the soil's not still performing. I've seen plenty of soils that don't look all that great, but I'm still getting some level of performance from the soil as far as nutrient cycling and the plants getting what they need. Our almond farmer, when you look under his soils, it's really hard to ever find a nematode, or very rarely we'll see protists and not that much fungi. But in reality, that farmer was able to reduce their nitrogen inputs by 80% and their potassium by 100% as far as reductions are concerned, and phosphorus by 50%. So is that a win for the farmer? Hell yeah. Is it great? No, there's still room for improvement type of thing. And I think Keisha, the thing that you would really hit on, which was if they're doing just light amount of disturbance, the soil might be able to kind of find this kind of equilibrium, where it's like it's not highly disturbed, it's still performing, but it still could be better in that sense. And I think we see that all the time, to be honest with you. And this soil is not disturbed. I mean, this soil, it was really, really crappy soil when I first started. And I'm 3 years into this journey of rehabilitating the soil. So it still has a ways to go. But, seeing an organism like this is not freaking me out especially because I have the context behind it.
I love that. What else are you seeing in the sample, this tree sample?
Let me, zoom out and see. I think I got some fungi in here. Let's go take a look at that.
Are those several nematodes there, or is that?
No, that was just some plant matter.
Oh, okay, got it.
Like some root
So
matter.
here's a piece of a fungal hyphae. You can see that there's some aggregation in here.
Mm.
Um, just looking around, there's some aggregates in here. Although this soil, from a standpoint of, is it a really healthy looking soil, like really well aggregated? No, not at this point. Um, but I can tell you from the context I also had from when I first started, there was almost no aggregation. I mean, this came from a field, or basically my front yard, that was really growing only early weedy species, heavily compacted sand. A lot of people think of compacted soils around clay, but boy, you can find sand to be heavily compacted as well. So this was a really, really, really poor soil. And the way I've really reconstituted was to use a lot of woody mulch and compost extracts and running plants through the system. That's what I've been doing for the rehabilitation. So
Yeah.
within 3 years, I went from almost no aggregation, almost no life in the soil to, it's okay.
Don't move,
But
Brian. Do you see the testate amoeba?
yeah, right there, he's a
Man
little
pointed it
hiding.
out.
Yeah, look at that.
There's a testate amoeba back over. Yeah, right
Right
there.
I
here,
missed it
Oh
in the
yeah,
chat.
nice.
right there.
Good eye,
Gordon, I
Brian.
don't think it's empty just because, I mean,
It's hard to
you
say.
can see it's got some
Let me get,
stuff inside. You can't
there we
clearly
go.
see
Yeah, I don't
anything.
think it's empty either, Keisha.
Yeah,
It
no.
looks like it down in that bottom part right down in here.
Uh-huh.
There it looks like there might be a vacuole in there. Hard to tell because there's stuff kind of around it.
I love its shape too. It looks like a little vase.
It does, doesn't it?
It's really beautiful.
It has that kind of papusa kind of thing to it. So, when I look at this soil, am I ecstatic about it? No. But knowing again where it was and where it's heading towards, I can live with it. All right, but we wanted to see some really, really crappy soil, right? So this is from one of our clients, and this is some agricultural soil. And this was taken from a field that is heavily tilled and very heavily fertilized, and about every aside you could possibly think of, they use. And let's just take a look at this soil comparative. So let me
Already
zoom
looks
in.
horrible.
Yeah.
Yeah, I
Looks
mean,
terrible.
when you get right down to it,
It's like sand.
it's just like sand. Yeah, it's just a lot of mineral particles in there with very little aggregation at
There's
all.
a little bit of aggregation,
A
but
little tiny bit, right?
it gets worse than this, you guys. This is actually like a
There's
3 on a
so
scale of 1 to 10.
What,
Yeah,
what's
look
your
at that.
what's your dilution on this one?
This is a 1 to 10 dilution.
Okay.
And we're right now at 400x total magnification. So thanks, Raleigh, for that, because that is a good point to
Will you
Will
point
you
point
out.
out,
check it?
So,
Brian,
Will
will you
you
point
check
out those
this
teeny little
for the
aggregates?
yeah,
oh,
aggregate?
sure. So, like right down here, you can really they're kind of easy to tell because of the color they come. They kind of get those tan to light brown to darker brown colors.
Amber.
And when you look around at the mineral component, a lot of other things are very shiny, sharp, and that kind of thing. Aggregates will tend to be a little bit softer, and that kind of deeper, richer color to them. But yeah, there's an aggregate right there, and there's a little bit of an aggregate that's got a little bigger chunk of mineral to it as well. But I mean, really, in the really sad fact is, I would say out of the most of the soils that we assess, like agricultural soils, this is what we see, or like Keisha even mentioned, even worse than this. And it really kind of sucks too, because we'll do maybe like 30 samples will come in, and you're like, oh my gosh, I gotta look through 30 samples of this. Where when you get a look at compost, you could spend 10 times longer assessing compost or really good soils because there's so many more things that you have to identify and see and be interested in. Like here, I can quickly look through this sample and go, oh yeah, I can't see much anything in here. You throw in a sample that's got a lot of stuff in it, it takes a lot longer.
Well, it
But
also too sometimes you're running a sample and you find like in good soil and you're just like, oh, this is awesome, and then just blow your whole test because you forgot what you were doing and just like following around and like looking
so
for
many
people.
times, you're
Or it's
like,
like
what?
4 hours later, I'm like, what did I just get hourly for that?
Yeah, no doubt, right? It did not pay off. Yeah,
I don't
but
know if
it's
this
you're
job
right.
is
It's like
paying off.
I've never seen that before. What the hell is that? And you're like, oh my gosh. And you got to get your camera set up and then record it and do all this other kind of stuff. And then yeah, exactly. And then the
And then
sample
deep
dries out.
diving
You're
on
like,
Google Scholar, just trying to figure it out for days. Yeah.
Well, I
oh,
see
too funny.
bacteria.
Yeah, this is
Yeah.
great.
Well, and this is one other point to look at this. So you look at this soil, and I'm just going to show you. It looks like it's vibrating, right? It has a motion and movement to it, and you have all these little small particles. Based on this dilution, you can't really tell if this is bacteria or if this is clay. And I know some context. This soil is from a clay, silty, mostly clay kind of soil. And so you look at all these little particles, I would suspect that a lot of these in here are actually clay particles. And why are they moving? It's because of a thing called the Browning effect. It has to do with water tension. It causes a natural vibration or movement amongst very, very small particles. And so we have to dilute this significantly. In fact, I would probably dilute this to 1,000 or 1 to 2,000 to try to get very few amount of particles. So then I can really look at the particle and say, is it smooth, round like a cocci bacteria, rod shaped like a bacillus, or is this rough and jaggedy like a clay platelet or some other kind of mineral component? So, all right, Keisha, why don't you show us something that has some life? Because this is wild.
Sure.
See some
Let,
good stuff.
let's see. Can I share?
I might have to stop sharing first.
Oh, shoot, sorry. Stop sharing.
Oh man, one thing this would be really cool if like with the sample we could show like the picture of where we collected the sample and then before we
Oh
load
yeah.
up the sample, that'd be a
That's
sweet
good.
thing. Ooh, hello.
Oh,
Oh,
Keish, can you can
hey.
you, can you go up to view and do the full screen?
Oh,
Can I go to
nice.
view? Where is view? I see no view.
Oh yeah, look at that guy.
We got it. We got one. We got
Our
Mr.
mascot decided to
Tardigrade.
appear.
Woo.
What's behind Raleigh's
I
head
feel
there?
stressed now about this whole full screen. Is it just fine if I leave it where it's at? Like my heart's gonna full screen.
Sure. I mean,
Oh,
Yeah,
like,
that's fine.
All
Casey,
right,
if you
Anthony's gonna
can,
come help. That's gonna make it better.
okay.
Full
Yeah,
screen.
if you click view full screen. Nice. Was this in a catalyst compost sample?
Oh, no, absolutely not. Tardigrades do not survive thermophilic compost piles very well.
Hmm.
Sometimes they'll make their way in like after everything cools off, but I don't think they have enough of a protection to hang out. See?
Yeah, no, it's
Okay, I was getting
it's
stressed
it's
about
not
it. Like,
there.
like it's just not
I have no
available.
idea what's going on. It's not available.
Yeah,
Sorry,
Keisha's gonna be like,
I can't
All
help.
see, I
good. All good.
told you.
We can deal with it. It's all
That's
good.
not an I told you so. I'm
I
always
think I
nervous
think the good
about.
thing is like everybody, you could zoom in on your own screen, like because I'm on a Mac and I can just click zoom in and gets around that.
Okay.
And
I
quick
love seeing
question.
those guys.
Stop sharing.
Jen saying, will leave access to the recording? Oh yeah. Yeah, everybody's getting it. We're gonna send out a replay after. I'm gonna
Yeah,
try to get
unlike
that.
the last microscope one we did where I forgot to record it. So this one will be official.
Oh, it's all good.
Oh
No,
no, that's so
I
weird.
feel like it's okay though.
It's fine.
Just
We'll just deal with it.
that's some good questions.
Yeah.
all right. So tardigrade, what is this sample? This is a compost sample that is my food waste, which is I have a lot of food waste right now, and my garden waste, which is just super diverse, lots of veggie cuttings and scraps. mixed with a handful of catalyst compost every time I dump my bucket. So what I've been doing is just taking all my garden scraps and putting them on the right. When I dump my bucket of food waste, I put a little bit of our compost and then I cover it with the waste. And the black soldier flies are just like taking it apart. They're amazing. Working on playing with them during the summer season because they just seem so much more efficient than the worms at making my organic material microbes. But so this is a really active pile. It's fresh. This is kind of a little bit of the goop that's left over. It doesn't smell bad or anything like that, but you can see if I focus away from the tardigrade, it's a really busy sample. This is also at 1 to 10. Um, so I'll back up a little bit. I'm at 100x total magnification there. Now I'm all the way out. And I might even want to dilute this sample so that we can see more. But this is what an active compost pile looks like. I remember Elaine telling me one time, I said, when should I start looking at my compost pile under the microscope? And she said, wait till it cools down. It doesn't make any sense until then. And so I've been looking at active compost piles for a long time, just trying to learn about them. This one isn't hot. It's at a really low temperature. That's a rotifer right there. You'll see tons of rotifers in this compost pile. I've got it pretty moist. The black soldier flies seem to like the moisture to take apart the woody materials. But you can see there's just a whole lot going on. Some really weird fungi over here that's super massive.
Oh yeah, look at that.
Yeah, I'm not parfocal
Got a
right.
chunky, beefy, stocky guy there, that fungi.
Yeah, excuse me while I try to find it. My screen and my screen are not the same. Crazy. Okay, I'm gonna try to find it like this.
And you're at 100 total, Keisha?
Yeah. Yeah.
Okay.
Let's see if I can get it. Now I'm at 100 actually. I was all the way up. Yeah, that's weird. That might be organic material and not fungi, right?
Oh yeah.
See how squiggly it is?
Oh, it does that curlicue thing. It looks like some cellulose. Yeah. It's like a bent
This
up,
is about
twisted piece of
100,
cellulose.
100x.
Oh
But you
wow,
can
that's
see some
huge.
fungi in there, right here. Cellulose, here's one. Oh my gosh. A lot of people think these spiral things are spirochetes. They come in all shapes and sizes. They're just static springs in the soil or in your compost. Those are just from breaking down organic materials. But see how busy that is? You can see there's two tested amoeba. It's like, where's Waldo in a sample like this?
Right.
I could probably focus up and down and just find so many different organisms. Some of them possibly being omycetes, because we're not controlling this with temperature. Um, but yeah, you can see right there, there's a flagellate coming through.
Well, I
That too.
spy two te amoebas or testates.
Yeah, the testate amoeba, that's a big guy too.
Yeah, there's also a lot of nematodes in the sample. Let's see. And some really interesting testate amoeba that I don't see in my other compost. But here, let's check out this nematode really quick. Go. Looks like a bacterial feeder to me. Can't see its mouth part very well on my screen, but you can almost see the valve right here.
Oh, yeah, right there.
It just pumped a little bit too there.
Yeah.
Yeah.
Yeah, it's having a snack. There's a lot of the, like lots of nematodes in here, super, super active.
I
All
got a
right.
question from the Q&A here really quick.
Oh,
What
yeah.
combination of computer, PC, Mac, operating system, camera, and software are you all using? So I think Brian is on PC.
Windows, yeah, Windows 11.
Keisha's on Mac, the newest version, whatever state park that's named after. And then the cameras are BioVids. They're BioVids from LW Scientific, but I think BioVid's its own brand, right? I don't think
It is
LW.
BioVid's its own brand. Yeah, I get my stuff from LW. I'm using the 1080 HD camera right now, my old camera, because my 4K camera is not working. I'm gonna have to get a new one. And Brian is using the 4K
Yeah,
And
floating
there's a few
bombing.
different, there's differences in these. I think Brian's has an H or has a Wi-Fi
Has
adapter on it.
yeah, Wi-Fi adapter, LAN adapter, HDMI, USB connected, all that kind of stuff. Although mine's broken as well and the USB is not working, but I connect that through LAN is what I'm connected to right now.
Yeah.
Yeah, and then
And we
the
got these a long time ago, right? I think I've had
Old.
mine now for
They're old, and I've toted mine all over the world.
5 years.
I took it to
Yeah.
the jungle last year, and I think that's that crazy fungus that's growing inside of it. I think it came from Costa Rica. Like, I shouldn't have taken it where we took it, but a really cool function though with the Wi-Fi adapter because we were actually like in the jungle looking at samples, and there was people on their iPhones around us that were able to tap into
Right.
Starlink and then watch what we were seeing on their phones while they ate dinner. It was pretty trippy. It was really cool. But there's a lot of ways that you can look at microbes with a microscope. Almost any microscope is capable of having an eyepiece with a cell phone adapter on it. Everybody has a cell phone. It takes good photos. You can buy old ones really cheap that are still high resolution. Um, the triocular is best if you have multiple ports. This port up here would mean that you could put your cell phone on it and then still look with your eyes. So that's kind of nice. But I know people that just have the one, and they put their phone over it and then they just look at the scope through their iPhone. Actually, the guy that makes the t-shirt that Casey is wearing does that. It's very cool. I would note here some different sizes and shapes of testate amoeba. This is a testate amoeba here. Very, very dark, and you can see a lot of texture on its test. I'm no expert in testate amoeba, but I have been studying them a lot lately, and I'm tending to think this one probably uses particulate from the soil to build that test. When you
Mm.
see it look irregular and mottled like that and really dark, they tend to be adding things to their test as they go. Down here, empty testate amoeba, someone was talking about that earlier. Is that empty? I would say this guy is empty. And look how
Yeah, it looks
tiny it is compared to this big monster up here. Um, Tested amoeba come in all shapes and sizes. You got to investigate it on your own. Go to someone in the text, Casey, www.arcella.nl. Do yourself a favor, go look through there, spend like 10 hours. This guy has spent his entire life collecting images of protozoa and then filing them into their categories. And it's just incredible the different shapes and sizes that you can see in water, in compost, in soil. They're all over the place. And they are epic nutrient cyclers. It's just like Brian was saying, you saw we're seeing so many amoeba in one place in the sample. And you see Brian's tree sample, well, we found one in the whole like slide as you were moving around.
right. Yeah.
It's still
Not
producing
many, right? A lot of
many.
times in soil, especially soil that's starting to kind of repair itself, it's hard to find protists and things like that. But looking at compost, if you're not seeing a lot of protists in there, then you probably got some problems with your compost.
Oh, and here I want to point out some stuff. These are things that people like are always asking about. So this circle here, this like this O, and then possibly this one here too. That's like a leaf stoma. It's part of a plant. It's a, you'll see these little rings when the leaf is decomposing. They'll be all over the place. Like I said, this is compost just like still being broken down. Up here, let's see if I can get it in focus. These pointy bits, these are all over the compost over and over and over again, probably another plant part. All of this dark material, this is all aggregation. And you can see humic and fulvics building up in it. I'll show you this sample. You see how dark it is? It's crazy compared to even our compost. This stuff is just building up humic
Oh
acid like
yeah, look at that. That was really,
nobody's business. I mean,
yeah.
I'd like to compare, when it's finished, I'm going to compare the humic content of like the worm compost versus the thermophilic versus the soldier fly larvae because it seems valuable. But yeah, what you're looking at is just tons of organic material in the process of becoming microfood. So the bacteria is breaking all this stuff down, so is the fungi, it's sticking it together. And then these protozoa that we're seeing, these nematodes, even the tardigrade, they're eating the bacteria and fungi and the protozoa. So this is a really awesome ecosystem. I'm super excited about it. Um, I guess my point I would like to make here is that when we're composting, a lot of people think the thermophilic compost is like the heat is what makes the microbes, and it's very much not true. These microbial ecosystems are all over us, and you can grow microbes in probably a million different ways. So this is just a new one I'm playing with that I've never explored before. I'll point out this testate amoeba that's reproducing at the top. That's
Hey,
really
Keisha, there
exciting.
was a comment in the chat around, are these little insects, are they fertilizing the soil? And I think you just described that it's not insects, but it is microorganisms. Well, there could be some insects in there, but microorganisms. And what Keisha was talking about, the bacteria and fungi and all that, that's nutrient cycling. That's how nutrients get cycled inside of this particular medium here.
Yeah, exactly. They are. I think Elaine, not Elaine, but Elaine, her best friend, coined the term the poop loop. And
Yeah.
I think it works out great if you can just imagine these little protozoa, kind of like you would imagine a cow or a horse. You want fertilization for your garden that's organic, you get poop, you put it on your garden, it provides fertilizer. And that's really what these little guys are doing, only they're just so much better at understanding how to operate at the complexity that nature has designed. They're so much better than us. And you can see here, like, that's one species of testated amoeba. Here's another, here's another, here's another. There's spores, there's fungi. All these, thousands of species of organisms live together in this just com really complex, diverse, minute, teeny tiny ecosystem. So, what is this, Casey? 1/2000th of a drop we're looking at right now?
Yeah, around there, right? I think it's 2150 or something like that of a
Ah,
single drop of water.
yeah, it's crazy. All right, Brian, let me know when you've got something. I can keep cruising.
Yeah, I can take over. I got a wetland sample.
Ooh, that's exciting.
But for
Those
everybody,
are
like,
always
I love how
fun.
this sample just by the top level, you compare it to the other sample. The other sample literally just looks like these tiny vi vibrating massive pebbles, like it's a desert. And this literally looks like you're looking at a forest.
It's
Like
live.
Yeah.
yeah, there's like giant predators there. There's
Yeah, it's like
organisms.
what is this thing?
Yeah,
Huh?
it's like
Yeah.
if you're in forest, like elephants coming through trees. Oh my God, what is this giant
Well, I
arthropod
can tell
leg?
you there are bacteria in the photo. There's protozoa. There's some fungi, and I don't know what the heck that is. It's probably a plant part. It
Yeah,
could also
You
be
know,
an
and
insect part. I don't
Maybe an
know.
insect leg.
yeah,
This is
maybe.
the wild, wild world of no temperature, my friends. It's way more fun than thermophilic compost.
and here's the thing. I mean, we collectively have probably well achieved well over tens of thousands of hours of time under a microscope, right? And thousands of samples. And there's always, I mean, there's so many times when I come across something, I'm like, I have no freaking clue what that is. I don't know. I've never seen something like that before. And that's quite frequent. I'll come across something like that. I mean, not in the agricultural soils, but something that has interesting, that has life in it. So just the amount of diversity that's out there is insane. All right, let me share out. Okay, share my
And
screen.
for Keisha and Casey, Shauna was asking about like references for okay, microscope, computer, and camera. Maybe we could list a few of those.
Yeah,
Wait, oh, what is
sure,
this?
Casey.
Oh,
Okay,
oh,
let's see, folks. We've got this is a wetland environment. So this is coming from a wetland that's in a forest. Mike Miller Educational Trail. It's semi-old growth around it, but then there's a nice wetland that's in there. And I mean, obviously, there's a really good tested amoeba right here. You can see that just really good structure, good, and it's not doesn't look to be appear to be empty. But look at all these diatoms that are up here. Crazy. We always see that when I look at wetlands, just massive amounts of diatoms. And this soil was pretty saturated, pretty wet when I pulled it. But as I'm going through here, there's actually quite a few tested amoeba. Let me see if I could. I had a slide up earlier or a field of view up earlier. Ooh, that's a good one. Can you guys see this one right here?
oh
Where
yeah,
I have my
love
cursor
that.
around?
Yep.
That little test date.
So what's kind of neat about that is here's the amoeba down the bottom of the test. But a lot of times when they get, think of like a hermit crab. It's a protective. It's a shell that they're able to kind of get into. They'll take material like organic material or pieces of aggregate and they'll plug up the hole that they have where they come out. And then when this testate amoeba is ready to pop out, it'll push that plug right on out and then go out and do its thing. And it would Keisha's had some really cool videos of the tested amoebas. So when the amoeba comes out of the test, it has these what they call pseudopods. Basically, it's I don't know how you describe the structure, but it's just it will put part of its body and just ooze its way out, almost like a finger or like an arm, and then it will wrap itself around whatever bacteria food that it wants to eat, and then consume it and pull it into its body. When you get to see them doing that, it's really, really cool. Naked amoebas, you get to see it because they don't have a test. They just always kind of ooze through the soil. They're a little bit harder to find sometimes because the test is very easy to identify. But yeah, I love seeing amoebas. Let's see. Do I have anything else in here? I had a really large arcella in here. Let me see if I can find that.
Good looking algae.
Oh, there's a flagellate. Can you guys see the flagellate? I'm tracking it.
I can see him.
Oh yeah,
He's
Oh yeah, there he
moving.
is.
He's moving.
He's right next to your little cursor, actually. That was close.
they are single-celled organisms.
There he goes.
There he goes.
Oh,
Like almost in focus.
he keeps coming in and out. They're super hard to
It's a
They are
great
hard to
sample,
track.
on
Brian.
video.
Loaded with
See
life.
what?
That's a great sample. It's loaded with life.
Oh, it's yeah, massively. Look at this testing amoeba right here.
I'm guessing it's not diluted, Gordon, because it's water sample, but is it diluted actually, Brian? Did you use
It's
milk?
not a water sample. I took it from a really muddy soil. It is diluted at a 1 to 10.
I
Um,
always dilute one to
Probably
10.
could have diluted it a little bit more. But yeah, we could see a lot of things in here.
Oh, the algae's still
Wait,
alive on
wait.
the
Is
left.
there something moving on the left?
Something's moving that around. You'll see that a lot where an aggregate will move and you're like, what's in there? It could be a naked amoeba, it could be a flagellate, it could be a ciliate. There goes the flagellate again, another
Oh, yeah,
flagellate.
coming down from the top.
And you can see the flagellum. I don't know if you guys can see it very well on the computer screen,
Oh yeah.
but this has a flagellum out in front as it moves.
Oh, there it is. Wow, the diatoms.
There's so many diatoms in here.
Wow. They're so
And
pretty.
it's a really healthy wetland that where I pulled this from. So it's one of my go-tos whenever I want to go see a lot of, cool, interesting things. I'll go to the wetland and pull it.
What is the role of a diatom in the ecosystem? Like,
Well,
what organism
Diatoms
is it?
are algae, so they're
Ah, got
photosynthetic.
it.
And they are able to produce that glass house that they live in. So it's a like silica.
Hmm,
silica, yeah.
I
So
see.
it's
It's
yeah.
organic glass.
Yeah, kind of neat,
They
huh?
produce oxygen, really. I mean, they're like, I think okay, so I don't,
I
I'm
got
really
it.
not great with numbers, so I hate to just spout anything off the just top of my head, but they produce a huge amount of the Earth's oxygen. If diatoms were to disappear, like we'd be in big trouble.
I think, like, honestly, I think it's like 70% of the world's oxygen comes from diatoms.
Casey loves spouting
Wow.
numbers. He's like,
Yeah. He's a numbers guy.
Yeah, they
the
outdo all
numbers.
the trees on Earth like 3 times over.
Yeah, their functions.
Well, thank you, small friends.
Yeah, you can tell this wetland's doing its job, right? It's got plenty of diatoms in there.
Yeah.
All right, Keisha, you got anything you want to share?
There's a question here, and I think
Yeah.
like great to stop and look. What are those things that look like fungi? I'm seeing some septa, and those are algae too. Sometimes algae have those kind of. Brian, can you
Oh,
like
right
move
here.
on to
Trying to
Right here.
try to find
spell.
one with some color in it so it's more,
Oh, there's one right there.
yeah.
There's two of them right here. There's like this one right here, and then there's this one right here. They have
Yeah.
that, yeah.
But those are individual cells of either a cyanobacteria grow that way. And
Yes.
there's also types of algae that grow that way where they actually they grow it in cells and they like chain together. And so it does kind of look like fungi, but if it was alive, you can actually see each individual cell is completely separated by its own cell wall. So it's not really a septa.
Is the wetland sample from a more or less aerobic source? Jeff asks.
It is definitely more in the anaerobic kind of way because when I pulled this sample, when I dug down and pulled it out, you could get that kind of sulfury smell that comes up from usually typically waterlogged or saturated soils. But to be honest with you, when you're looking through here, it's not that bad. I mean, I'm not seeing I haven't really even seen any ciliates yet. And I'm seeing plenty of tested amoeba in there. So I think this is in a zone that is the wetland is starting to dry out. And I took it from an area where it was probably underwater maybe a month ago, and now is in this slow dry out period. So it's probably pulling in and infusing quite a bit of oxygen. If I would have gone farther in, and got a little bit more really wet kind of soil, I may see definitely would see something a little bit different.
Veronica had an excellent question we should touch on before we move on. I thought algae sucked oxygen, as in the like the algal blooms.
Huh.
Um, no, well, anything that's photosynthetic takes sunlight and carbon dioxide and produces oxygen from that. Um, but I think the suffocation of the fish, I don't know. Yeah, it could be through just reproduction, right?
Yeah,
Because
Casey, I
they,
thought it was
they use oxygen to reproduce.
yeah, they reproduce so fast or yeah.
Yeah, basically they're reproducing so fast that they're using up the available oxygen, but they are also producing oxygen as a gas because they're photosynthetic.
Well, I mean, also just like the reality of how many different species of algae, diatoms, cyanobacteria are there, and the algal blooms, like typically those are coming from, first of all, like an overabundance of food resource, right? Like those are happening not in like necessarily the most natural of systems. It's when there's a lot of nutrient that's flowing and they're able to just like go gangbusters and grow on it. It's a good question though. They
Yeah,
do.
I wish I knew more about aquatic systems. I need to to get that the organisms and everything else down a little bit better.
Sorry, so
We we had a
I
few
looked it up.
good
I looked it up just to see. So
nice. Get some
it is
facts.
true. Yes, they produce and consume oxygen, but they only produce oxygen during the daytime when they're photosynthesizing. And so at night is when they deplete the oxygen supply.
They're
Ah.
good and bad.
And bad.
They have it at nighttime is when they come out and do their naughty work.
What
All
was it like
right.
the Gremlins, right?
Yeah.
Gremlin.
Yeah, we had a few good webinars on like dealing with dead zones and lakes and ponds, and I think a big thing is people basically floating wetlands are one way to clean up these giant algae zones. Because of course, fertilizers is the big issue here. So much of it is just flowing into all our streams, which goes into our rivers, which cause massive dead zones and then spreads to the ocean. But like upland and in ponds, people can kind of do floating wetland systems that can help these out. Um,
I had something, but it stopped moving. So,
Mm.
all right. Yeah, Keisha, anything you want to start sharing?
Sure. I've got some, let's see, I have some vermicompost from our first class that is more finished. It's not in a state of active decomposition right now.
oh, Keisha, while we load this up, should I tell folks about Compost Academy
Oh
real
my
quick?
gosh, you are on, with everyone's on the edge of their seat. They want to hear the
Everybody?
stuff.
They
Let's
do.
go.
Well, folks, we're going to take maybe 2 minutes. I'm going to tell you a bit about what we're doing with Compost Academy. I mean, it's so great seeing the folks that we already have who are in it, who've gone through it. I mean, we just did a webinar with like Domingo sharing his stories about Pacific Northwest composting. That was a really awesome one. We can send out the replay afterwards, but so many people who are a part of this webinar right now have been through it and are starting to make their own compost, sharing the experience with their gardens and their farms and their compost lots. It's really extraordinary to see so many people here. But for the folks who are new inquiring about it, basically, we have started this program called Compost Academy online. And what we're doing is we're offering online training. Oh, sorry. Can you guys see this all right? Or is it the presenter view?
Yeah. Yep. We're good.
Okay. On how you can make compost that actually that will give you that you will be able to create these like nice, mature, biologically focused compost. So when you look under a microscope, you actually see what looks like a forest. You see fungi, protozoa, nematodes, and you don't just see these sands and bacteria and clay particles. Like we want to give you the tools to actually make really high biology compost that you can actually restore soils with. So, you go through these webinars, you get inspired, and where you go from here. We want to give you a program that could actually make you feel experienced to produce compost that can actually get you a result on your land, that can help you get better yields, that can help you push succession forward basically from a desert back into a grassland, back to a productive farm, or even further. And to give you guys some case studies, this is what Keisha and Casey have done in Southern California using the catalyst compost and using multiple applications of extract. Really awesome case studies. This is just after a few years of applying compost extract. And these are post-fire landscapes where this was just ash before they started. And by using biological compost and extract, they were able to push the succession further and further and further till native species are coming back, till greenery is there year round. It's really extraordinary to see these up close. And you don't just see plants, you see keystone species showing up, deer and owls and hummingbirds and butterflies. It just keeps going on and on, the benefits that you see from using biology in these landscapes. And just a few more of these, like just the change you can see by using biology on the landscape when you make the right compost is amazing. It's so drastic. It can go from a berry ashen shrubland to closed canopy in several years by knowing how to use biology in the right way. And like Brian's had amazing case studies using this on vineyards and orchards. So it's not it's landscapes, it's farmland, it's marginal land. Anyone can use this, whether it's a garden, farm, even an urban lot. Like there's no end to what you can do by harnessing biological compost. And just a few more case studies. These are a few things from Hollywood where really you have the heart, like really hot urban landscape that was turned around with applications of soil biology in just a few years. So, we created this program called Compost Academy to show you how you can get the same results. And that's by training you practically how to make biological compost. So it's about camaraderie. You're going to be able to do this with a big group of people. You're going to be able to form a team and action plans. Keisha, Casey, Brian, and sometimes I might chime in and help you coach you to the way you feel competent and confident to do this. You're going to have action challenges and experiments where you're going to be pushed to actually do this and not just learn theory. And you're also going to be able to do it at your own pace because we do offer self-paced training as well. And of course, we want to help people with professional networking and educational networking. Whatever we can do to help, we're there. So, what you're going to learn, wide spectrum composting techniques, thermophilic composting, vermicomposting, static. We're going to show you about how to use biological extracts and teas, brewing, scaling, application methods. The microscopy part, though what we're doing is kind of an intro, we are going to have some microscopy sessions. We're going to show you how to replace expensive and toxic inputs with biological compost. And we've got lots of case studies about people who are landscapes, farmers, and growers. And of course, we're going to dive deep in the soil food web and expand on kind of the lessons of Dr. Lane Ingham. And of course, we want to show you about how to compost and the scale you need, whether it's small or large scale. So, a bit about content. We have 28 lessons. These are the self-paced program. We have 13 weeks. We've got training. We've got an online community, Microscope library. Got action challenges to push you guys. Great discounts for future workshops and access to really cool upcoming intensives like the large scale workshop coming up in California in October. So you get four of these programs for the price of one 13-week coaching program, self-paced program, the Soil Circle community, and the Soil Explorers group. And if you want to jump in on this and join this community, there's a lot of good benefits here. I think it's a value of around 5,000 compared to like what other online schools are offering. And really, this is altogether, it's only $1,100. You get free lifetime access to the self-paced training programs and the workshops recordings. There's going to be bonus sessions like this that we're going to peppering in. And you get a $1,000 discount on the future Compost Academy online, which we're going to film later in the year and launch in 2026. So if you want to take part in this, we got an early bird offer available till July 15th. It's only 1,100 bucks, and you can sign up right now at Academy in our first 2025. This is our second program, coaching program that starts July 31st if you want to hop in. So I'm going to pop that link in the chat box and hope you can join us and join a really awesome contingent of people. Some of the folks from the first round are going to join the second round. They're going to give feedback. They're going to jump into challenges. So it's going to be a really great group, and it's got amazing results from people already, and it's just going to keep going further and further and get better and better. So with that, we can get back to sampling, but we'd love to see everyone who's not part of it already join the Compost Academy and join the group.
We had the question come up. It always comes up. It should come up.
Mhm.
How are we different than the Soil Food Web School training? Good thing. Gordon already said something I was going to ask. We've got a bunch of students that are in the chat right now that are actually Soil Food Web graduates that are also in our school that can speak their own opinions, which is always great. And we need to get some of those out because we've got recordings of the most lovely
Yeah.
feedback from folks about this. But you know what? It's different in so many ways. I think, first of all, it ties together really well. I'm a student of the Soil Food Web School. So is Brian. So is Casey. We actually started at,
There's
uh,
Raleigh.
Oh
and
yeah,
so
we
is
all
Raleigh.
were.
Yeah.
Yeah,
Raleigh's,
we all
Raleigh's the
we
OG.
all.
He filmed that stuff.
Yeah.
But what was it called? Environment Celebrations Institute. So we were in that original iteration that had, its ups and downs, and then the new school. And I can say that our school is different in the way that we're just really active with you. It's more about community and working with your specific reality. Like we talk about nutrient cycling, we talk about nematodes, we talk about microscopes a little bit, we talk about compost, we talk about how to organize your life around composting so it fits in and it's a little bit easier to do. Lots of garden planning and working on how do we work with customers, how do we work with farmers. How do I look at my entire year and understand how much compost I need to treat X amount of land with this much tea? We've all been working in this industry for, well, Casey and I have been in ecosystem restoration for like 15 years, but we've been working with microbes for a good solid 8 years now. And I think we're just really excited to share what's working. I mean, Brian is working at scale for many, many, many of those years. He's got lots of experience he can share. Casey is a magician at working with large scale equipment and transitioning it into what you need to actually apply microbes. And I'm super passionate about helping people figure out their niche and like how they're going to make money and enjoy it. So I think there's a lot of ways we're different, but we attract Soil Food Web students. So you'll be with those people if you join us.
Well, and too, it's like we're about the actions, the challenges, the practical experience that, we helped when we helped the lane, like Soul Food Web was, there was lots of theory with a little bit of practice, like practical how-to on there. And we wanted to be like, okay, let's switch that up. Let's do a ton of practical experience learning and also touch a bit on theory, but not get bogged down in it.
Oh, Dan, that's a good brag. Dan's like,
Yes.
he's like,
I know.
boom.
7 350-gallon compost
I
piles.
know. That's epic. Good job, Dan.
And
Whoop, whoop.
Impressive. All right, let's get back to my
Back
sample.
to the same.
Hold on. I feel like this sample looks like there's lots of microbes in it, but I feel like it looks really lonely compared to the last sample I showed you guys.
We should also get a sample that's just like the most polluted, disgusting sample we can find. Just maximum spirilla, maximum roofing,
Oh,
like a
yeah. Oh,
sewage
yeah.
treatment plant. That would be interesting.
Maybe we could do, where you can find that is like if we're ever on a job, Brian, where they have, we know where they flood the fields and
yeah,
they're dropping the nitrogen at the same time and they have those algal blooms
Oh,
in the ditches.
right.
Those are the
Yeah,
most interesting spirochete, spirilla.
totally. Manure lagoons too.
Yeah, yeah. Ooh.
Ugh.
Oh, Raleigh, you got a manure lagoon across the street.
oh yeah, I do. I do have a water treatment plant. I should just like take a
Sample.
scoop.
Head over there. Yeah.
Yeah.
Grab some.
All right, this is a compost sample. It was again, no heat on this one. It was made with worms from my food waste from the spring. It's just now finished. It's a little garden now. I am at 1 to 10 dilution, and I am at 100 times total magnification. And so you can see here, testate amoeba, testate amoeba. Here's another big Arcella, another testate amoeba. I like the view from here. I think it's a lot faster and easier to gauge quality. I'm really into microbe adventures, especially if you're not a person who's doing a scientific study. I think it makes a lot of sense to spend as much time as you can exploring these ecosystems. And this is kind of like a bird's eye view where you can still see organisms and understand what they are without being so zoomed in. So I'm just going to move around this kind of freely without looking through my eyepieces and see what comes up. And you can see it's just not as busy with organic material as that last sample was. Now you'll see a flagellate. It must be really big if we're seeing it at that.
Magnification.
Yeah. And you can see aggregation. Another testate amoeba, I believe, right there.
Gordon had a before we move on. Gordon had a question. Is that a spore near the aggregate material at the bottom?
Did I move too much? The
You
one that
just
looks like a
moved.
You
football?
may have.
One that looks like a little football?
Yeah, yeah. I think that's what
Oh
Gordon
yeah,
was
to the
saying.
bottom
On
right.
the right now, just move over a little. Yeah, the little football down on the very bottom right hand side. I think.
That's a fungal spore, I would
Yeah.
say, probably. I could look closer. Some yeast have that shape too, but they usually have a bud scar. Um,
There's
yeah.
so many different shapes and sizes of fungal spores. It's
Mhm.
amazing.
Yeah, even like especially like mycorrhizal fungi and I mean just so many. That's another thing to just have fun Googling. There's all these charts out there of just different fungal spores and shapes that you can look at.
There are people, that's their profession, is to simply identify fungal spores. They'll spend their whole lifetime getting into
Or
that
PhD
fungal
on it.
spore came from this type of genus. Yeah.
Ooh, that's
Yeah,
a
and then
nice little
only to
fungi.
specific families sometimes, not like all fungal
Right.
spores, just like one family of or one genus of freaking fungi. I got an interesting question here from Veronica Alice is when the prey dies, does the necromass nutrient become plant nutrient available, or does it only become available through the prey predator system? And I don't know, I like that one because it's like it is actually like the necromass like being eaten, it's still being eaten. It is part of the prey system. So when something dies, fungi, bacteria, protozoa, even animals, on a microscopic level, yeah, it's not you're not going to be seeing ants or anything eating it, but it's bacteria and fungi are eating that and consuming it, and it does become available nutrients in the soil. It's still part of the predator prey system. Their prey just happens to be dead already, and it's bacteria and fungi that are eating it.
Yeah, I mean, I think like my brain goes both because I'm imagining like okay, there's a nematode and a huge whatever flatworm comes and just like claw bites it in half and like the flatworm's gonna poop out that nematode, the part that it ate, and that is gonna be available nutrients for plants. But the part that's still kind of like dwelling in the soil decomposing, I think. My brain's thinking it knows that the bacteria and fungi might consume that and it might be tied up in the nutrient cycle. I think it could probably go both ways.
Yeah, I think it's probably mostly coming from the predator prey, but I'm assuming when cell walls get punctured and so forth, there's proteins and things like that that get released that are going to be, available for the roots of a plant to be able to pull up nutrient cycling wise. But yep, predator prey is probably the biggest part of it. Anything that's going to be broken down is going to be eaten and consumed and recycled. And Keisha, there was a question around, is that an empty test over to the right?
Looks like it.
Looks like it's empty. There's a flagellate dragging that thing along.
Yeah,
Looks like
Yeah,
an
that's definitely an empty test right there.
nobody's home.
That's fungi. This is like some people will send me these pictures and say, "Is this an oomycete?" Because it's not perfectly, uniform diameter. It is a perfect example of like maybe a bump or a place where the fungal hyphae just chose to grow around something,
When
you
you
know.
shook out the sample,
Or
it
got
got
squashed,
Sort of
banged
squished.
up.
pushed
Yeah.
it. Yeah, some fungi you can see it as you look at them. They're very thin and they'll fold like tape does over and over again, and so they won't look so much
Right.
like, it looks like it's really damaged. Yeah. And then this, like, this is another one. People send me this. What is this? It's a big, I don't know. I don't know what
The
that is
orb.
actually.
Imagine.
Brian, do you know what this is? What,
Let's see. Oh, that right there?
what's your,
Brown.
I get pictures of these all the time and I don't feel like, I'm like, it could be
Yeah,
amoeba. Look at the inside. There's more stuff inside of it than there
stuff
is
in
on the
there.
outside, but I don't know.
I don't
So
know.
where did the sample come from and what's the
This is the
dilution?
compost that I made during Compost Academy
Nice.
class one.
Okay.
This is your static pile, right?
Static
No,
pile,
this
yeah.
is your active pile. This is your active pile, yeah.
It's not active anymore. It
It's
finished already. I
right.
thought it was going to take a year.
It
I
wasn't
thought it was going to take
thermophilic,
a
it
year.
was
It's done.
static.
Huh?
Yeah.
It wasn't thermophilic, it was static.
It was static. Yeah, not
It was
thermophilic.
static. Okay, okay.
Yeah.
Yeah.
But it's mostly
Yeah.
decomposed. I don't, it's like, and I see these all the time. Sometimes they look a little more folded than this one does. You see
You
what
know
I
what?
mean?
That light color, I've seen it too, and maybe it's just you can't see it, but sometimes the spore sacs for fungal spores will be like that, and they'll have a split where a bunch of the fungal spores will protrude out of.
Yeah.
And that kind of the end part of that is that lightly colored kind of sac, but typically you'll
But
see the cut or the break,
every
but
time I see that, it's a circle, like Pac-Man.
oh, really? Like a full
I've
circle,
never
yeah.
seen it quite with this texture. Like, you see this right here? It's not a circle. There's something.
Right.
And then here, like it's, I don't know.
Yeah, it's not
Anyway,
quite
I'm gonna show
round
you.
round, is it?
Does anyone in the chat know? I can't see the chat.
Yeah.
No.
Those bacteria are trapped in there.
And then let's see.
And I mean, this is common, and we come across all kinds of stuff in the soil. We're like, I don't know.
And this is something I would like to show off too, is just like fungi being fungi. Maybe I can back up and you can see it better. There we go. So like if you were wondering like how does fungi create structure in the soil? It is just so, so good at reaching out and grabbing inorganic and organic materials and binding them together. You can see right here, this fungal hyphae is inside of this aggregate, and who knows how far it's worked through all of this. And you can see it. It might branch there and go out and just start connecting. So fungi is making these epic connections and creating these huge aggregates. Like, remember Brian's aggregate from the really poor soil? I had to like someone had to ask, like, can you point it out? Um,
Mhm.
in really rich soil, often the aggregates are so big that you can't really even see like here. It's like you get into it and it's just like, okay, now I'm at 400. Can't really even.
And that's the thing is you can zoom through it, right? Because you can see through a lot of the aggregates unless they're really, really thick. But don't forget when you're using a microscope to really go through the whole focal plane because you'll miss stuff that is not on the focal plane that you're zoomed in at right now. You need to
Okay,
go
see the
up and
testate amoeba
down
in
through
there?
that focal plane.
See it?
See what?
Testate amoeba, it's in focus
Yeah,
right now.
right.
And it goes away.
Yeah.
So like
There's
observe,
always cool things sometimes. You're like, oh, well, look, there's some fungi
well,
right
yeah, the
there.
idea of being able to like that the human eye can see all that there is to see in a sample. I hope you guys can like understand just from this little blob that, how much time do we need to take to really understand what's inside of this aggregate? We cannot understand everything that's living in the soil from one soil sample, but also look at everything that we can see. We can see this aggregation happening. We can see the test data maybe. We can see, we get a lot of data, but I think this shows the limitations of it really well.
Ethan keeps asking, he's asked a couple of questions about endophytes, and I'll do the more simple version here. Do you see a correlation between the number of endophytes and the microbes in your soil?
Yeah,
Hmm.
so if there was an easy way, like what we need is a wand where we can like an endophyte wand where it's like you are looking at a plant and you take the wand and you touch it to the plant and then you go, tell me of your endophytes. I don't think there's an easy way to know what species are working with the plant currently, what function they're playing, what level they are. I mean, Brian, do you know of a test where we can actually understand endophytes in a clear way?
Not an easy readable or a non kind of like university style kind of test. Like going out to agricultural fields, we don't typically do that. You could probably, well, because you're going to be finding it inside the plant material itself. when we do plant sap or plant tissue, we're not accounting for those kind of things. So there's no good test that I know of from like a commercial standpoint to really understand the level of endophytes and the kind of endophyte quality that you have inside of your plants. I mean, from scientific research, we know endophytes are hugely beneficial. They do a lot of different things for the plants. They're great allies. But being able to really see, or quantify the level and the value of them, yeah, I don't know from a commercial standpoint.
Yeah, I'm also the idea that some organisms are not, they could be an endophyte or not. So whether they're acting, in that way, in that moment with the plant, these
Right.
are these crazy specific interactions that go on in the soil. And I hear this more and more. Like I feel like our work is like it's like everybody wants to further complicate it. And I get that because there's so much exciting information out there. I mean, if I spend a couple hours on a white paper, just digging through white paper lands, like I really want to get specific as well, but the complexity of this is so ridiculous compared to what we can actually test for.
Yeah,
Sometimes it feels better just to go for diversity and try to use the microscopes and the tests we have just to, gain some sort of
for
baseline.
sure. And I think there's kind of an educational knowledge gap, from what the science is coming out and what we're starting to learn and understand about the role and the complex world of the microorganisms in relationship to the plants is enormous. How that translate into practical and how you do your work to kind of establish those kind of really strong microbial ecosystems, there's a huge knowledge gap that's there. And I think that's what we're starting to figure out is when we, find scientific research, and then we start doing our work, how do we try to mimic that so that we can take advantage of some of those learnings? I think there's a lot to be still kind of figured out.
And there's one big giant testated amoeba.
Look at that.
Big
It's
boy.
nice and
Oh, is that a spore? Spore there.
there's
Like a
some fungal
cluster of
spores down below.
spores. It looks
I
like they're
don't
all
know if it's
bunched
fungal spores.
together.
I think it might be pollen oriented in a funny way.
I like Mark's comment. The inverse is just as complex as the universe.
The innerverse.
Yeah. Yeah, I try to simplify over. Yeah, I don't know.
This
I
might be a
would
good
like everyone to know where we're
opportunity.
at. We don't know much. We're explorers.
This might be a good opportunity. If anyone wants to ask a question live, we could unmute you. Just raise your hand in the Zoom panel, and then we could ask a live question. Otherwise, we can just keep going on samples because there's a lot of cool
I'll
stuff
throw
in
up
there.
something while you're waiting for a live question to
Yeah.
come in.
Don't be shy, y'all. We want to get some questions in.
I'm gonna share one thing really quick. So
Oh,
Ed
shoot.
Tevnan, I think, he put in like in a question in here asking about
Mhm.
he's doing a talk or something in his hometown and he lost his microscope, and so I was gonna post his contact info back into the chat to see if anybody's near him that could maybe rent him a microscope or something.
Nice. There we go.
I just put it in the chat.
Yeah, New Hampshire, I think is where he said he
Yeah,
was
he's
coming
in
from.
New Hampshire, so anybody in New Hampshire, contact Ed
East Coast.
there.
So like Keisha was just showing about, fungi inside of an aggregate. This is from my vermicompost, this is from my worm bin. And you can definitely see as you go through that the focal planes, how well integrated that the fungi is. And if you take your time, you can kind of go through. Like here's another example. I don't know if you guys can see it right here,
Um,
right where my cursor's at. So when we assess and we look at these things, you want to make sure you take your time. I mean, really, with just a fraction of me moving my focus knob, it would go in and out of focus trying to look through this aggregate. And you can also, you know, turn up the brightness. A lot of times, if you want to see through an aggregate, you really can turn up the brightness to kind of get some kind of light coming from below and through. There's all kinds of things in these aggregates. And this is something, that's really important for us to understand, especially as practitioners. The value that, aggregation gives to us is it does a number of things, but one of the biggest things is providing structure in the soil. pore spaces so that we have water holding capacity, we have an exchange of oxygen, inflow of oxygen into the soils. Soils that don't have lots of aggregation or that soil structure tend to be heavily compacted. They don't infiltrate water very well, don't hold water very well, and air doesn't flow through that system very easily. So by having bacteria and fungi, which are the prime drivers of creating aggregation in the soil, they're able to pull and create these aggregates for us, which is it's just amazing.
I got a question here that Keisha's gonna love. Do any microorganisms translate to beneficial gut bacteria?
Yeah, of course. I'm gonna love that. I was yeah, I mean my story is that it did. The reason I got into composting and the reason that Elaine's words even spoke to me were because I was obsessed with food ferments. Casey and I were living on this crazy mountain in the middle of Ecuador, like way far away from roads. We didn't have power or refrigeration. And so lacto fermentation, fermentation, dried meats, cured meats, all these kind of things, cheeses. That was really what made our world go round. So we were obsessed. I was obsessed. Um, with harnessing wild microorganisms and using them to our benefit to store food. And so it always made sense to me that you could do this with soil inoculums. And as soon as I came back to the US and saw that they were indeed selling these teeny little packets of microbes for like 40 bucks, I felt like, yeah, I had to get into it. And for me, the compost lot and my digestive system, like both happened, like my digestive system turned around when I started focusing on our compost lot. And it's not like I have went to the doctor and had them stay up on my microbiome and how it's changing over time, but um, yeah, healthy soil has literally like changed my life. And having my hands in compost all the time. I know a lot of your microbiome comes from your mom. It's like a really substantial part of it that you carry with you your whole life, but there's a lot of interesting studies out there that talk about how you can receive parts of of other people's microbiome when you shake hands or hug. Basically, as you move through the world and bump into all kinds of things, your microbiome is changing, and sometimes the microbes stick and dominate, and sometimes they don't. And that's just another like infinitely complex reality that we live in every day. But I think there's a reason why, if you're depressed, they say, go take a walk in nature. It's not just that you're taking a walk, it's that these microbes don't just have to do with your guts, they have to do with probably every single function in your body because we are one with them. Microbes made the soil that made the plants that we eat. So, yeah, I think it probably has a huge effect. Okay, done with my
Great.
passion speech.
So this is where, like, when we have to assess soils, it becomes, or in this case, we'd be assessing compost. It can take a long time. I mean, just looking at the screen, there's quite a few tested amoebas in here. Plus flagellates you got to look for, fungal hyphae. There's definitely some cool ones in here.
All right, so we got some EM questions coming up
A
too.
lot of EM questions coming up. I had a question for Jack. Was it Jack that brought it up?
Yeah, Jack brought it up.
Jack says, recently Matt mentioned using EM1 in compost and it would keep material from flashing off by reducing oxidation. What material is flashing off? What do you mean,
Yep.
Jack?
And
Jack?
he also referred to more complex humic compounds developing and becoming more stable. So the one thing I think with like all of that is like EM1 is like what is in the EM1? Like what bacterial species are we talking about here? Like 99% of the time what you're seeing in there is some form of lactobacillus, which basically you could get out of it's in all of our fermented foods, it's in cheeses, it's in yogurt, it is a probiotic, they're facultative anaerobes. Um, But with most of these EM1 products, like if you look at the back of the bottle, you'll find some type of lactobacillus, and you will find those exact same species that already live in the compost. They're already there, they're already present. Like adding them over the top of it isn't necessarily, and it might make those numbers, increase, but at the same time, it's it's I don't know. I see it as like, I don't know. It can be effective if the compost is already really bad. Adding facultative anaerobes to like an anaerobic compost is going to make that compost better because those microbes are good at regulating that anaerobic activity. But if you're producing a high quality compost already, I guarantee you have not just one species of lactobacillus, but you could have hundreds of species of lactobacillus. And isn't that always going to be better? But I think a lot of it comes down to whether or not the compost you're producing is actually any good. So it can seem like it's saving really bad compost, but really good compost, I can't see a real reason to be putting that on there.
Yeah, well, okay, so I think so. Astrid just said, I think EM is a food source for other protozoan. Effective microorganisms is another term for bacteria. It's a bacterial species that they're working with. So they are food for protozoa. Absolutely right. Um, and EM, I like what Casey said. I still don't fully understand what Jack was a asking, but when we are talking about the EM, like, okay, so Matt Powers is, I send him our compost all the time. He's a big fan and he does a lot of DNA analysis on it. And you guys, we've got a lot of students of Matt in this group, so you guys should ask him, or I could text him right now just to make sure that I'm not putting words in his mouth. But I'm almost 100% sure that he was like, your compost has EM in it already. So that's one reason that I don't like necessarily add anything to our compost. The other reason is I feel like with the product that we make, we've already accomplished our goal. And a lot of the work we do is around looking at what is your goal that you're trying to, like, what are you trying to accomplish? And then getting there without spending too much money, too much time, making it too, you want to do your job. So for me, I don't know, I already put so much work into all of our processes, the idea of like going out, buying a product, making another product, putting it in a sprayer, spraying it on my compost, cleaning that sprayer out, putting it away, like, wow, just saying it out loud is a lot of work if I didn't have to do that stuff. So for the compost we make, I don't think we really need to add EM, but somebody's added a whole bunch of information here about EM, and it looks like maybe a copy and paste, but I'm going to need
Yeah,
to read that
Ethan.
before I speak anymore.
Well, Ethan has his hand raised, so I might
Oh
unmute
yeah,
him
bring
real
them
quick.
on. Woo!
Hey, Ethan.
Hey, how's it going today, y'all?
Doing
Good.
Hey,
good.
Ethan.
Good.
Excellent. I just wanted to go over what Matt Powers means by flashing off, and that is the evaporation of off gases of active byproducts and nutrients due to, sunlight, heat, aeration, exposure, and lack of adhesion time for our microbials in the soil or compost.
Okay, so
So like loss of nutrients through gas form.
yeah.
Exactly. Yeah. What's being released volatilely, like carbon dioxide, ammonia,
So
odorous
how is he
compounds.
measuring all of this to like what are his okay, so say I wanted to go and test this on my compost and say I'm going to make a windrow and I'm not going to use EM. What data points is he taking and measuring? And then he's adding EM and taking the same data points and then finding that it was much improved.
So to be clear on that, I'm not 100% clear on how he's effectively doing that. I believe it's lack of what's present. So let's say you do a test without EM and it shows there's a huge lack of presence due to the flashing off and disturbance. But when adding the EM, when you see an increase for some reason, that's not just magic happening.
Yeah.
There's binding that is going on in that soil structure. There's enzymes being released, and there is an apparent reaction, very much like products that are inoculation products on shelf. If they don't have a timestamp of how long this is good till, you can kind of tell that they're adding a solvent or something to keep it in a stasis instead of keep it active and alive.
Okay, so yeah, I'm still curious. So the metric, like what are you measuring here? That's what I'm curious about.
Is it like biological uh like
because it seems
abundant
to me like
or
you're
is it
saying there's a change in
nutrient?
the bio, like the microbiome, like you looked at it with a microscope and it's different, or perhaps you looked at DNA analysis and it's different, or were you measuring, like I know NASA has the capability to measure methane now. Is like that what you're talking about?
I'm with what I'm doing, it's working with Lloyd Dixon and just doing the test analysis of the aggregates, saying, oh, what's
Okay, so you're looking at
present,
aggregation.
aggregates and chemical analysis. If there's specific presence of X, then Y is happening.
Yeah.
That's the example. There again, just like endophytes, there's more testing that needs to be done to really signify the exact situations that are going.
Yeah, cool. Yeah,
Yeah.
I would love if you have more information that you could send. Yeah, I hate to try to speak on something that I don't know anything about. It would be yeah, that sounds like
Same, and when
way over
I
my
can't
head, Ethan.
same, and when I can't clarify how Matt specifically is doing it, I just know the flashing off term and what
Yeah.
he said about that on his webinars and
Yeah,
stuff.
for me, I need people to be able to break it down really simple because like there's so many of these terms that yeah, honestly, like they're being pulled from scientific literature and used so frequently, and I just don't understand. So I need that kind of stuff to be just like broken down in the most simple form so I can start to aggregate it and come up with an understanding, you know? But I'd
Absolutely.
love to look into this. I should write Matt. Maybe we could have a collaborative presentation sometimes and we could get into this a little more because I'm sure he can simplify it for us. He's good
Yeah.
at that.
So
Absolutely.
Jack's asking about that too. So potentially, maybe this EM treatment means that it prevents um, compost from releasing a lot of material as gases. Therefore, we just re
Yeah.
loses less material.
I think
Again,
maybe
too,
they're
I
saying
think
it's going to have more nutrient content if you're
Maybe.
using the
Right.
EM1. What I'm interested in is if the EM1 is already present,
if it's
how do we
already
test this,
in.
right? Like, so how would we test this at our compost lot to find out if it's worth it to start adding an EM1 product to our windrows?
Yeah.
That-
Because for us, it's like when we got like the DNA testing on our compost specifically, it was like I don't remember exact numbers of what species of lactobacillus and bacillus, basically the entire those two groups, but it was super high numbers of both of them already existing in there without us adding anything. And so yeah, I'm curious if oh, just need money for testing everything, don't we?
Well, and I bet, with a lot of these things, Matt could have been pulling this from some scientific research that's been done. But, when you start really unfolding the test process, what kind of compost did they use? Was this just some bulk compost that came off of kind of an anaerobically
Municipal
produced
waste.
and exactly. And, it'd be really fascinating to see if there was a difference between the different types of compost, one that's very biologically rich, one that's very biologically poor, and see if that's a difference, right?
Hmm.
Because there may even be almost no difference adding it to a compost like Catalyst compost, where if you go to just a typical bulk compost, yeah, then maybe there's a huge difference in that sense. Interesting. I'd love to see the paper though. Keisha and
yeah,
Casey, if you guys get the information,
I have
I'd
to
love
reach
to
out with
see it.
Matt.
well, yeah, I'll hit Matt up and do a little bit of talk. I'll talk to him about it and see. Yeah, I think what Ryan's saying, like as far as I've heard it, I know EM1 is used a lot in low quality compost, and it's going to help. I mean, it's going to
Helps with the
help it break
stink.
down so much faster. Almost any bacterial jadam and like any of those, like any bacterial soup is going to help it, especially with moisture. And Carl's saying, while composting is beneficial practice, it's crucial to manage off gases like ammonia, methane, nitrous oxide, volatile organic compounds to optimize compost quality for agricultural and minimize environmental impacts. For sure, Carl, right on
Yeah.
point with you.
Most of that stuff, though, is produced in anaerobic conditions. So it's like if you're trying to minimize those environmental impacts is by keeping that compost aerobic and keeping it actively turned, making sure you're monitoring that, not overwatering, not letting it sit for too long at high temperatures. It's like our compost lot is in the middle of a neighborhood. Like if our stuff was smelling like this and smelling really putrid and, of that ammonia smell and those things like that, we'd get called on all the time. So it's like a lot of the times with these super massive, large scale facilities is they're just piling, they're just trying to reduce the waste. It's not really about the end product at all. The end product is not the important part. The part is getting the materials in, making it small and getting rid of it. So It
I was
just
like, remember in Costa Rica, they're like, oh, this is our go-to. I'm not sure if it was like they're making their own bokashi, but this is our compost, and it just smelled like
everybody was
just
gagging.
pure
And
vomit.
gagging. Yeah.
And it was like,
No, I
don't
mean it
worry,
doesn't
this is
have
great.
it doesn't have to be that way, but at large scale, it really is like, I mean, it depends on what we're talking about. We're talking about growing microbes in compost. If we were talking about waste reduction industry, then EM1 would be a really good addition to it. I think it's low cost. It's going to help. But yeah, even we don't produce those sort of gases in the compost that we make. I mean, I want it home every once in a while, but
Probably
never
small amounts. Probably small amounts. I don't think there's any way to
tiny
avoid it.
small amounts. But even remember, we're working with like some of the biggest dumps in California, and you know how they actually measure all of those different compounds is with human nose. Like we are the most sensitive at smelling them, so you know when you're producing ammonia and methane, it stinks.
Ethan raised his hand again. Go ahead, Ethan. I think you're still on.
I was curious with the anaerobic side, are y'all working with any bokashi composters, making bokashi compost for any companies and doing the anaerobic system that way that produces those high ammonia volumes?
No.
We don't like the smell. We're like, I'm all about pleasure in my work, and I just hate that smell. It's like, I know it's not fair. Everyone's like, you should do bokashi. It's amazing. I just like don't. It's gross. I don't. But other people do bokashi, and I think it's great. I have some friends
Sure.
that are
Would
very
make
into
it
it.
smell good with EM1 and let's say baker's yeast or even brewer's yeast that are in the supplemental departments.
Yeah,
Yeah.
great. That's great. I think different strokes for different folks. There's EM people out there, but yeah, I don't.
Sure.
Yeah.
Yeah, and that's the thing is there's no like one right way, to be honest with you. There's so many different ways to be able to do this. There's plenty of wrong ways to do it as well. But yeah, like Keisha, we had that one client that was a K&F farmer, rocked it in K&F, and it was like, all right, great, that's your gig. You go, you have it dialed, you're working it, right? Unfortunately, just some people will just take that and say, well, that K&F's the only way to go. No, and I don't think that we're ever going to say that either.
Yeah, no. No, it's just like what you like. I think that's what's kind of neat about our program is there's ways to do everything. It's like even when you're talking about bokashi, I've smelled bokashi that's like, oh wow, that's like a ferment or a baker's yeast. That's not so bad. And I've smelled bokashi that like literally like almost threw up in my mouth. So there's a right way to do it, I think. Dujon's a guy, he's using KNF and bokashi almost primarily for all his food scraps and garden, and he does incredible things with it. So
Thank you for answering all my questions. I really appreciate it.
Thank you for
No problem, Ethan.
Thanks,
participating.
Ethan.
Gordon had the slimy bokashi too. Yeah, for sure. I like it's so interesting how some people it is the microbes that they're cultivating and they're anaerobic. Same with food though. I can remember when we were first trying to get like raw cheeses down,
Mm.
we'd capture some fungi and bacteria that were just so gross.
From your stocks.
Oh, yeah.
Brian, this is excellent with testating me, but what is it?
This is my vermicompost
Nice.
for my worm bin. Yeah, just testing me was like crazy in here.
Mhm. Cute little orange ones or yellow ones.
Yeah, some yellow ones in there. That's some really, really cool ones. My worm bins are doing well, and a good, decent amount of fungi in here too. So I'm happy.
Tons of
Look at
tests.
that color.
Jack says another can of worms.
Nice.
It's like another can of nematodes for us. I think we should keep a running list of these, these big questions. There's been a lot of them that come up where we just, not a great answer, but we spent a lot of time reading through white papers and talking to experts and compiling our ideas. And gosh, the
For
jumping
sure.
work was a big one. And we had a few of them. Structured water was one everybody got excited about, but
Okay, go ahead.
you know, just compiling all these questions we have and waiting until we
Here's
can.
a good question, kind of a follow-up. It's important to remember that while the sniff test is a valuable tool, it's not foolproof and should be used in conjunction with other indicators like compost temperature, texture, visual inspection, material. Any thoughts?
I think you should be a
Um,
teacher, Carl. You know what you're talking about.
yeah, for sure.
You're right. You
Yeah,
shouldn't just use one indicator ever.
right.
No.
Yeah, one of the things we talk about, if you don't have a microscope, one of the ways to assess compost is your nose, obviously, is one of them. Your eyes, visually, the color, and then the feel, the touch. How moist is it? Does it have stick to it? There's the texture that comes along with it. You can gain a lot from those three senses as far as the quality of that particular compost. Is it foolproof? No, not by any stretch of the means. You add more tools like a microscope, you get a better assessment. Get a better you want to do DNA analysis, even more granularity data. So you can add more data points into it to kind of really narrow it down. But boy, those three, your eyes, your nose, and your hands can go a long ways.
I like that you added texture too. Most people don't observe that, but there's a stickiness, a quality to a really a microbially rich environment that like you can feel it with your hands, actually. Jack says, are nematodes more prevalent in vermicompost? No, I wouldn't
No,
say
I
that.
don't think so.
I would say nematodes are more prevalent in young compost or in places where there's just a lot of food resource.
Yeah.
It's
Especially
hard to,
like a fresh active like very like new like maybe one turn in on a thermophilic pile, your bacterial feeding nematodes go
Off
absolutely
the
nuts.
charts.
They go nuts. But it makes sense. That's what the heat is. The heat is bacteria reproducing, right? And so it's just become a feeding zone for them. So yeah, that's the time I've seen the most nematodes ever in any sample is like first turn active thermophilic pile.
My most was, do you remember when we had that gray water system on our yurt?
Oh, yeah,
And
yeah, yeah,
I had those like
yeah.
sunflowers that were like, oh, they're so big. I was like, maybe I should look at it under the microscope.
I
And it
forgot
was,
about that.
no, there was, you couldn't count how many nematodes per drop. All
Almost
this
like you did a nematode extraction without doing a nematode extraction, right?
without a
It was
nematode
just
extraction. It was insane. And then you know what? As soon as it dried up, they were all dead. I went and looked at it again when it was just, I think it
right.
wasn't that it dried up. It was that the sunflowers stopped growing and they turned to seed. And they're
Yeah.
just, it wasn't, the vibe wasn't right anymore. And the nematodes are all gone.
Hmm.
Oh, look at that diversity of testing amoeba in one view.
Like that one. Yeah.
Eek.
All over the place. Isn't that crazy?
Good work, Brian.
Yeah, this is really cool. It's always fun. Test amoebas are just one of those neat things because there's so many different shapes and sizes, and yeah.
Big one, small one, some as big as your head.
Rod, we should write one. There is not a good book on identifying all of the organisms. Matt Powers has a book on on microscopes, and it's got some identification stuff in it. I want to do one that's just on microbes only, though, and it's in the works. Got lots of photos and lots of plans.
Here was a pretty, I think it was kind of an intro question, kind of an intro science question from Citros, saying too much bacteria and fungi in the human body equals sickness. Questionable. But what about the soil and how to control them, considering that they play a vital role in the soil? So I guess,
I mean, it's not our expertise, the human body, but I assume that like the human body is out of balance, just like our soil is. You know, your microbiome is destroyed as soon as you take antibiotics, and same thing when you put asides on the soil. So yeah, we
yeah.
go.
And Ethan, follow-up question here. Have any of you heard of y'all heard of Jonathan with Capture Tech? He has a pilot program to reverse toxic algae floating biochar rafts. Ooh, that sounds really cool.
That's neat.
Kind of
That
No,
makes
sounds like
sense. I've never heard of that though. Yeah.
yeah, I mean like there were some other cool yeah, what we did were about floating wetlands to control toxic algae. So it's like floating biochar wetlands. That sounds even cooler.
or I'll ask another compost question, basically about yeah, you guys can read it if you want to. It's just like if the compost is still warm to the touch, like should you use it or should you send it to a lab? If the compost is still warm to the touch, definitely steaming, don't use it. It's a very bad idea. If your senses have told you that the compost is still hot, for goodness sakes, don't put it on your garden. Let it cool off first. It's still active state. I wouldn't waste money on chemistry analysis until it's all cooled off. Um,
I would say too, though, it does depend on the time of year a little bit. Like if you have like we'll have our compost sits at like 86 degrees typically, like even for like months and months after it's finished, it's just ready to go. So in the middle of winter, you stick your hand in there and it kind of feels like you're touching a body. It's kind of weird. It's like just slightly warmer than your hands are. And that's okay. Like that's warm to the touch, but if you stick your hand in there, it's hot. Like hot to the touch is not good. If you're seeing visible steam, again, though, that is relative. If
Yeah.
it's 32 degrees outside and your pile's
Sure, if
steaming,
it's freezing
but
outside and it's steaming,
if
it's a
it's
40
if
degree
it's
pile. But
summer and that pile
Right.
is steaming, do not put that anywhere near your plants. You're gonna bake them to
Yeah.
death.
I had a
yeah.
client that they had bought some compost. For my first visit out to them, and they said, "Hey, we've been using this compost. We've had it here for about 4 months. Can you take a look at it?" And when I walked up to it, it was black in color, just completely black. And I opened it up, and it was about 120, maybe 130 degrees temperature in that pile, and the smell came off of it. And I was like, "You've had it here for 4 months?" Yeah, we've been using our garden, but and one of the reasons they called me is like, this is not working, it's not effective. And I'm like, yeah, well, this is probably one of the reasons why.
That's what
Just
the large scale facilities do. They heat it up for 14 days and then they just send it out the door. They don't
Right.
care.
Yeah.
It's like
don't
it's about
care. It's all about
making
flow
that product
flow
flow.
through.
Yeah, they've
Flow
got
Yep.
through.
more waste coming in the other side that they've got to get rid of.
Get it
That's
out.
crazy.
Jen asks, I'm on LA City water and there's chlorine and chloramine. Are there ways to still use water from the hose and do regen ag and make compost?
Yep.
Carbon filters.
Yeah, you can get an inline carbon filter. I think what's that guy's name? The
Boogie Blue
Boogie
is
Blue is
common.
one.
Yeah.
He's cool. We've met the guy that actually made him, so he's fun, interesting. But there's a few different brands. It basically screws onto the faucet, screw your hose onto it. It's about that big, and it's just a carbon filter. It's not going to take out everything, but it's going to help for sure reduce the load.
I will tell you, all of the compost that you saw today was made with chlorinated
City water.
water from my house. I do not use a filter. I do not want to spend extra money. I do not care. It is a rental, and you saw my biology. So we might just be repeating that because someone told us that it's true. I'm sure it kills some of the biology, but like, come on.
Yeah, it is also funny too, right? It's like you think about the idea of like neutralizing, say, chlorine and chloramine in a bucket. You'd use humic acid, right? That's what would neutralize those chemicals inside of a container. What is compost if not just like an insane amount of humic acid, right? Like if you can take compost and dunk it in water and make that water better, can't you then just put that water on? Like, yeah, okay, you're probably going to lose some, but if it's compost, it's full of humic acid. It's going to bind up that stuff. So it's not going to be able to take it all out. And it's something that's constantly being generated in a compost pile as well, right? So it's like, you might bind up some of it. And then when you go to put more water on it again, it's going to bind up even more. So yeah, I don't know.
Not use chlorinated water at Catalyst. It's we do
We
care
don't
there.
use chlorinated
Don't
water at
care at
Kettle.
home.
That is not
Yeah.
chlorinated, but
Well, and I think there's this variability. The closer you are to the water treatment plant, the probably the higher load of chlorine and chloramine you're going to get.
well, yeah.
And there's a water quality is an issue. We did with the agricultural space, especially with the deep wells. We get wells that are 2,000 foot plus deep, and the water is extraordinarily salty. Doesn't look like chlorine, chloramine, but we have to address that. Okay, that is a potential problem that we have to look at. So water quality is a thing, but also I think it might get overblown. Like she just showed you, using tap water out of her house. Can you produce compost with it? Hell yeah. Really good compost.
Yeah. Yeah, I mean, Brian, I would say that what you're talking about, the wells are way harder to work with than chlorine and chloramine.
Yeah.
Um, Jonathan's well. Oh my goodness, the salt on that thing is insane. It's
Yeah.
way harder to treat. You can't just use it for extracts. It kills everything. I actually like I'll see if his water isn't being treated and I make extract with it, like everything dies within a few hours or less.
Mm.
Hey, there's a ciliate.
Nice.
He's just crazy.
Well, it looks
You
like
guys see it?
yeah, yeah. Well, we saw some floating around. He wasn't like zooming. I know some ciliates are. This one is
It's
like
right here.
bumbling.
That's the ciliate right there.
Oh yeah, he's kind of like vibrating and shaking.
Well, you see ciliates, we call them race car drivers because they zoom across the screen, but when they're
Yeah.
grazing, they're gentle little things that just kind of move around and they graze and they eat,
Hmm.
Right now, that ciliate is just eating away.
Mutual. Ooh, what's a so that's cool. That little corkscrew at the bottom, that's got to be cellulose, right?
Cellulose.
Yeah.
Yep.
Mhm.
It's a disease. Run!
Ah.
I have
Run!
one of my favorite stories is when I first started doing this, I went up to Elaine's place up in Oroville when she was part of the ECI. And there was only three of us, and we were kind of going through this class. And told Elena, I said, I found the most amazing fungi. Look at this thing. It's corkscrew. It's uniform. I can't see septi in it, but man, this is the funkiest looking fungi I've ever seen. Nope, that's cellulose.
You're losing your sample.
Yeah, it's starting to dry out.
And just quick mention, like one cool thing about when we do microscope sessions during the academy, it's like everyone joins as a panelist with Kajabi, so people get to share their own samples, versus Zoom's kind of limited, where unfortunately we don't get to share each other's samples. Yeah, I see
Yeah,
that.
Keisha,
Jeremy.
anything else you wanted to share?
I think we're I think we're there. We're past
We're
there.
over
Yeah.
time.
Oh, hell
I
yeah. Look
would
at that. Wow.
I would
Time
love to
flew.
stay on the microscope with you guys for the rest of my life, but I got stuff to do.
But hold
Yeah.
on, look at that perfect amoeba inside of its test right there, straight down the middle.
Oh yeah, yeah, yeah. It's
Right
Yeah,
insane.
here.
you can see the amoeba really well
Ooh,
inside of it.
nice.
If I can.
That's very neat. It looks like it decided to.
And the one below it.
Yeah. This one right down here.
Yeah, awesome.
Well,
Let's
awesome,
see if I
folks.
can.
Well, oh, go ahead, Brian.
Oh, I was gonna say, let me
One last hunt. One
I
last microbe hunt.
can't escape out of this. Interesting. Cool. All right. Well, that
That was funny,
was
guys.
fun, guys.
For showing
Yeah, it
up.
was a great session. We'll get the replay on YouTube ASAP and give everybody a little replay email sent out today. And we're trying to get Joe, Vivian, and Todd on next week. Like Joe was down, I'm still waiting on final confirmation from Todd and Vivian. So fingers crossed I'll get a yes by Sunday. If not, we'll think of something cool for next week. But hope everybody enjoyed that microscope session and hope the folks who are new, you had a great time and want to join us at the academy and hope everyone has an amazing week and that your gardens are pumping out. And we will see everybody soon.
Good to see
All
everyone.
right, ciao,
Bye.
everybody.
Hi, everybody.